absolutely my pleasure to be here today to talk to you about some of the work we've been doing. And I just want to start out with the critical part of the work, the people, because obviously I could not do this all on my own. And the work that I'm going to show you today was predominantly done by Samote Foliaki, and also Katie Williams and Brad Groveman. For those that are less familiar with prion disease, just a little bit of introduction. These are neurodegenerative diseases of humans and animals. And once symptoms have onset, they are fatal. The animal diseases, there's a few shown here. This is scrapie that affects sheep and goats. It's the oldest known disease, known since the 1700s. This is bovine spongiform encephalopathy, BSE, and the press lovingly dubbed it mad cow disease. This, unfortunately, it arose in the 80s, and by the 90s, it was clear that it had transmitted to humans, probably in contaminated foodstuffs. This disease down here is chronic wasting disease, and this is probably the most relevant one for North America right now. In terms of human disease, there are three etiologies acquired. I just alluded to that mad cow disease caused. It caused a variation of an already known disease called Creutzfeldt-Jakob disease. The variation caused this disease to affect people much, much younger. So a lot of the people who suffered this were sort of the 18 to 30 age bracket. It was very worrying at the time. Fortunately, in the end, it wasn't as many cases as we thought it might be. There's been iatrogenic transmission through blood products, cadaver-derived products, and contaminated surgical instruments. These are fortunately rare occurrences. And Kuru is also another transmitted disease, and it was a disease of the Foray tribe of Papua New Guinea. This actually taught us an awful lot about these diseases because it taught us that eating contaminated meat would transmit the disease. And the Foray tribe, it transpired, were actually practicing ritualistic cannibalism as part of their funeral rites. Fortunately, once they were told this is what was causing their disease, they stopped, and I believe Kuru is now considered an extinct disease. By far, the most common form of these diseases is sporadic, so we don't know why they arise. It's just unfortunate. It's like most cases of Alzheimer's. I'm going to talk a little bit more about those in a minute. We also have a hereditary component where autosomal dominant mutations within the prion protein gene causes a prion disease, and the phenotype of that is defined by the mutation. The average age of onset is around about 65. It does vary depending on the disease, so it's a bit younger than other dementias like Alzheimer's. Traditionally, they've been very difficult to diagnose, and diagnosis has really been based on post-mortem examination of the brain tissue, finding this spongiform change, which is mostly characteristic of the disease. And in fact, the other name of these diseases are the transmissible spongiform encephalopathies. We have got much better diagnostic techniques now, so pre-mortem diagnosis is, I think, mostly possible. There's no cure, no treatments, no vaccine. Treatment really focuses on making the patient comfortable. So a little bit more about sporadic CJD, because I'm going to be talking a little bit more about particularly these two types of sporadic CJD. This is the most common form of human prion disease. These are still rare diseases, 1 to 3 per million. But they have several subtypes under this sporadic CJD banner. I think it's 6 or 7. The subtypes are defined by different biochemical differences within the proteins and are influenced by a polymorphism at codon 129. That can either be methionine or valine. So you can be a homozygous methionine, homozygous valine, or heterozygous. In the population, heterozygous is the most common genotype, followed by homozygous methionine, and the homozygous valine is relatively rare. In most populations, 0 to below 20%. It is a polymorphism, it's not a mutation, so it doesn't cause disease, but it does influence disease. For these two subtypes of disease, you can see there's not much difference in the age of onset or the range, which is very broad, but there's a real difference in the clinical duration. A lot of people with type 1 disease will decline rapidly and die very, very quickly. And often they can die before diagnosis because it is so rapid. Much, much slower for the type 2s. They also have different presentations. The type 1 is mostly cognitive, but the type 2 here predominantly ataxia. So again, making it difficult for a physician to determine what disease they're looking at. In terms of our genetic diseases, I'm going to introduce you to two of these in particular because these are what we work on. Genetic CJD has been shown to be caused by a number of mutations in the prion gene. I want to say something in the region of 50 have been reported, but most of those are not very penetrant or may even be polymorphisms. One that is highly penetrant, almost up to 100%, is the E200K mutation. And the presentation of that disease is very similar to MV1 SCJD. Fatal familial insomnia is different. It's only caused by one mutation, and that's the D178N. And this has to be in cis with methionine at that polymorphism. So they must be on the same allele. If it's valine at the polymorphism, the disease is actually CJD. So the mean age of onset of FFI is a bit younger. It's nearer to 50 years old, still has a very wide range. The clinical duration is actually determined by the second allele, whether it's methionine or valine. Presentation is, as you might imagine, sleep disturbance. So these patients, they lose the ability to cycle through deep light and REM, so effectively not getting proper sleep. And when they do go into REM, they may act out their dreams as hallucinations and results in a confused state. So very unpleasant disease. All of these diseases are caused by prions. Prions are misfolded conformers of a normal cellular protein. That protein is anchored to the outer leaflet of the plasma membrane. It's predominantly alpha helical with 2 end-linked glycosylation sites and an extremely long flexible tail, much longer than is depicted in this diagram here. So during disease, this is the change it makes into this conformation. You can see it's a substantial change. Once this conformation is assumed, somehow this is able to recruit more of the normal conformation and add them to this ever-growing fibril. Eventually, there's so much that it starts depositing, and it can be diffuse deposits or plaque-like deposits that we see as these brown clumps within the brain. There's still an awful lot of unknowns in these diseases. We really don't have a handle on how this change eventually causes toxicity, neurons to dysfunction and die. We don't know if this has a corrupted function or a gain of function. We don't know if it's critical that there is a loss of this as it's consumed, or whether it's a combination of everything. We also don't know why we have the different subtypes. The different subtypes really characterized by different prions attacking different brain regions and causing those different phenotypes. We think it's probably small changes in the structure that encode that, but it's really not clear why some cells would be more vulnerable to that change than others. We don't know what the function of PRP is. It's been linked with many different functions, and maybe its function is a linker protein, but without having a really good handle on its function, it's really hard to say what it does when it dysfunctions. And of course, throughout all these diseases, we don't yet know how to intervene in a way that's actually meaningful for patients. So when I came out to Rocky Mountain Labs, I had just started trying to develop a human cerebral organoid model of prion disease. The reason we wanted to do this was because we didn't have a completely human model of disease. I want to say back in, I think it was the late 90s, someone published that it was possible to get the SHSY5Y cell line infected. That was never reproduced, and we couldn't reproduce it in our lab. And I think that's probably because that line is it's so clonal now. Every lab has a different version of that line due to different culture practices. So we wanted something that would give us a better chance of getting a fully human prion infection. When we started out, we opted for this. This is the Lancaster and Knoblich Nature Protocol for making cerebral organoids. They are miniature brain tissues that we grow in a dish. You can grow them from embryonic or induced pluripotent stem cells. We use induced. That's partly because we work for the government and embryonic work is generally frowned on. But the induced have a specific advantage that you can potentially get them from any donor who consents that has potentially any mutation that you want to look at. So that has been an advantage for us. They contain all the cell populations of the cerebral cortex that come from neural ectoderms. So that's populations of neurons, astrocytes, and oligodendrocytes that structure within these nicely together. And as I said, this is the protocol. It really is very simple. You make embryo bodies that start the germ layer differentiation. Then you start their neural induction, transfer them into matrigel droplets, which adds some structure for them, some support, and more nutrient. And then eventually you put them into agitated culture that allows better diffusion of nutrient. This is what they look like. When they're young, you can see these really highly structured domains appearing. The center of these is your most immature cells, and as they mature, they radiate outwards. So you can kind of see that's what's happening at 35 days. Really, by about a month, it's very hard to see them under the microscope. And a couple of months, they look like this by eye. So they get quite large. Obviously, it's a model. No model is perfect. There are a number of pros and cons. For us, the pro was that they are completely human background and they have that 3D self-structuring organization that allows us to look at how various cell populations interact with each other. They can be manipulated just like any other cell culture. You can treat them, you can harvest them. So there's a lot that you can do with them that you would be able to do with a cell line. And they have a very long culture lifespan that in the context of our prion disease model is useful. Cons are that they will never mature to be a fully adult tissue. You differentiate them through a fetal pathway. So although you end up with mature neurons and astrocytes, there's still a fetal signature to them. They do lack certain cell types. Anything that doesn't come from neural ectoderm will not populate. So microglia is one of those cell types. There's no blood-brain barrier either. We do see a fair bit of batch-to-batch variability, and we do try and account for that in doing multiple batches to make sure we're not just seeing something strange within a single batch. And they can be time-consuming and expensive, especially for how long we take them out. However, we persevered. We thought it was worth it. So I'm just going to tell you now a little bit about how we developed the models that we have. So starting out with the model of infection, when we started this, it was a little bit of educated guesswork. When would be the right time to infect them? Would they even take up infection? What would be the right route of infection kind of questions? It was published in the literature that about 5 months is when they have mature complements of astrocytes and oligodendrocytes. The neurons actually populate much quicker. So we thought those support cells would be important for infection and decided 5 months was the time to infect them. For the infection route, we overlaid them with brain homogenate from people who died of sporadic CJD. And we actually used an MV1 and an MV2 to do this because our organoid genotype was codon 129MV. They were heterozygous. Then we incubated them out 3 to 6 months to see if we could get infection. And I'm cutting the story really short by saying, yes, we did. We saw prion deposition in some of the organoids, though it's brown plaques that we would see in the brain. We saw seeding activity. Seeding activity is really a measure of how many misfolded conformers are capable of recruiting more. It's kind of your infectious units. We saw that for all of the infections. Initially, we weren't sure if we were seeing astrogliosis. We were only doing sections, but since then we've done many more infections, and we see astrogliosis in about 2/3 to 3/4 of the infections. Cytokine secretion is also changed. Again, that can vary depending on the donor infection. And what was really exciting for us is we seemed to see differences when we infected with the different subtypes. So now we thought we might be able to tease out how these subtypes are affecting different brain cells. But before we did that, we really had to prove that the model we had was actually an infectious model of the original prion that we put in there. We're really fortunate in the prion field that we have excellent animal models. And when you inoculate an animal with prions, it will become ill in a very defined, reproducible time period, and it will have highly reproducible pathology. So we actually took our CJD brain homogenates and our organoid homogenates with the equivalent subtype infection, and we injected them into humanized mice. These were the TG66 that I think expressed 16-fold the amount of human PRP. And we monitored for infections to see if our type 1 infections and our type 2 infections were genuinely different. We actually had to do a couple of passes for this because our organoid infectivity levels at the titers are so much lower than the brain homogenate. So I'm just showing you the second passage here. But you can see in the orange, there's the MV1 brain homogenate and the organoid homogenate, and they overlap perfectly. The MV2s are almost overlapping, and there's a huge difference in survival, indicating that we really have got different prions as a result of these different organoid infections. Also, when we looked at the pathology, we were able to see a different deposition pattern in the ones that got the type 1 brain homogenate inocula. It was much more diffuse. And those that got the type 2, we had these really quite strikingly enormous plaques, further supporting that we really are able to differentiate the two different subtypes. So from there, what we have been doing since is actually a lot more infections. At this point, we have done 3 independent differentiations using different batches of organoids at different times. So trying to eliminate any issue of batch variation. Under these 3, within each one, we did 1 to 2 infections with the normal brain homogenate as a mock control, the type 1 MV1 brain and the type 2 MV2 brain. So all of these results are going to be amalgamated here. We generally take 2 harvest points. 90 DPI was when we started seeing changes in their metabolism, and 180 we had a really robust infection. So that really became our harvest points. And just before I tell you about the results of that, I'm also going to tell you about our models of genetic disease, because I'm going to compare and contrast what we've found in these 2 models. So for the genetic disease models, CRISPR-Cas technology has been extremely useful. We have a number of IPS lines from E200K genetic CJD donors, and we've been able to engineer those back to having the control normal cell line. We also have a number of homozygous lines for the mutation. I'm not going to talk too much about those today, but that can tell us a bit about allele interaction. As well as having these donor lines, we've also used normal lines, and by normal I mean no known neurodegenerative disease. And we've engineered both the genetic CJD E200K mutation and the D178N fatal familial insomnia mutation into these. We also have a couple of other lines, a Parkinson's disease line and a Down syndrome line. And the reason for this is because a lot of work's been done on those, so they really provide us with a nice control. For the organoid models of genetic disease, we didn't need to do nearly as much workup. These organoids differentiate and mature normally. They have the same cell populations when they have the mutation, so if they don't have the mutation. We have no evidence of replicating prions just for the mutation being there. And that's probably not surprising, because this is a disease that onsets much older and our organoids represent a more fetal state. However, they are susceptible to infection if we overlay them with brain homogenate. The mutation shows no evidence of increased cell death, and in fact, actually some of the cell death pathways are downregulated, but we do see significant astrogliosis. So there is some evidence that these organoids are a little aggravated. So the research aims for all of these is really to understand the mechanisms by which neurons dysfunction and die during prion diseases. Ultimately, we want to be able to find a way we can intervene that is good for patients. And so I'm going to start out telling you a bit about what we've been doing, look at neurotransmission in the infected and the mutation organoids. We started out using our donor E200K organoids, and this was actually before we'd made the corrected ones. So our healthy control organoids here represented 3 just normal healthy people. We used multi-electrode arrays, where you can see this is what the array looks like, and you put the organoid over the top of it, and then you read its electrical output. The readouts look like these, and really the way to interpret them is the bigger the dot, which is each node of these electrodes, and the warmer, more dense the lines, the more active these organoids are electrically. We see a significant increase in activity from 3-ish to 6 months. So from here on in, actually, we really stuck with doing 6-month readings for most things, or afterwards, because then that was when the network was really at its most heightened. But you can see our E200K donors had significantly reduced activity. This was also true of the Down syndrome and the Parkinson's, which was good because this was previously published and supported what we thought it should be. When we looked at the FFI organoids, and now we actually had the corrected organoids, we found a similar thing. Now, these graphs, this is the spike rate. So this is kind of indicative of action potentials on those electrodes. The burst rate is when you get several spikes in one go, which indicates a local network communication. We found, particularly in the FFI organoids, the spike rate was significantly decreased. It is quite variable. This is because the organoids have to sit perfectly over the electrodes, and they do have a tendency to wobble. But we could also look at forebrain neurons that we engineered, that we differentiated from these engineered donor cells. And when we looked at those, we also found a significant decrease in the burst rate of those forebrain neurons as a result of the mutation, indicating that there is an intrinsic fault within the neurons. Looking at our infections, and so that what I'm showing you here, individual organoids from 3 different normal brain homogenate MV1 and MV2 infections. They're grouped together because we really don't see much variation between individual MV1 and MV2 infections. But you can see 90 days post-infection, which is in total about 8 months old, because we start at the 5-month mark. There's a significant decrease in their neuroelectrophysiology, particularly the bursts indicating neural network communication. And it's more so in the type 1s where potentially the disease would progress quicker. By 180 DPI, it's incredibly reduced in all of our infections. We've actually done this a couple of different ways, and it's really reproducible that we lose this network communication. Somebody looked a bit further at the E200K organoids, and what he found was that they were not at all responsive to GABA treatment, GABA being an inhibitory neurotransmitter. We did wonder whether that's because they're already really at their baseline. So he also tried a neurosteroid, pregnenolone, that influences the GABAergic pathway, and that actually was able to stimulate some kind of recovery in that pathway. From here, we did qRT-PCR looking at a number of receptors. This was an 83 gene panel, and we found substantial decreases in a number of the GABA receptors in our E200Ks. This is the Down syndrome here, which again has been reported to affect the GABAergic pathways. So that was nice that we were seeing what we expected. Looking at this panel of genes in the infected organoids, we actually had relatively few changes. There's only one gene that was significantly changed in both. And what I should say here now, this is all of the infections we've done, and each infection is normalized to its relative normal brain homogenate control, and it's representative of 2, 3, or 4 organoids averaged within that group. So in doing this, we're intending to remove any batch variation. And yeah, you can see that the only gene that's significant is the GABA subunit. The MV2 only had 2 other genes that were significantly changed, and both of them were GABAergic subunits. The MV1 infections had 8 that were increased and 4 that were decreased. And of the increased, it seemed to mostly affect glutamatergic neurotransmission. The MV2s were quite variable, but some of these outliers were not included in our statistical analysis. So both between the E200Ks and within our infections, we are looking at an imbalance of inhibitory to excitatory neurotransmission, which is pretty interesting. We actually don't have this data for the D178N FFI organoids. Those organoids we're currently looking at single cell sequencing on, but I don't have the data for it yet. What we do have for those is we had the forebrain neurons. And for these, we did 4 separate differentiations of forebrain neurons, averaging 3 wells of each. And we did that same neurotransmitter panel. We found 9 genes that were changed. This is most of them. All of them were increased. All of these are significant. And we see there's still a hint of the GABAergic pathway being affected, but a bit more glutamatergic pathway, glutamatergic receptor subunits being increased in these. There's a couple of interesting genes here. This cholinergic subunit and hypocretin. And the reason these are interesting is because they're both linked with conditions that affect you nocturnally or night and sleep cycle. So these actually might be feeding into the phenotype of disease. So that was pretty exciting. We were also able to look in the mutation lines at neurotransmitter levels. We did this by mass spec, and Samote and Benji were able to set this up. So here, this is actually our control lines, our mutation lines, and then we've got the homozygous mutation for each of these, for the carrier, and our engineered lines, our engineered FFI. And I hope you can see straight away across the board, we have a reduction in GABA detected. We also, interestingly, have an increase in acetylcholine in the D178N FFI organoids. That, to me, is especially interesting because cholinergic neurons are suppressed when you sleep, and GABAergic neurons are mostly suppressed too, but there is a population that wakes up, and they function to keep those cholinergic neurons suppressed. So again, this shift in ratio of these two neurotransmitters may promote an enhanced state of wakefulness. So GABA and glutamatergic signaling are very intricately linked. They can be taken up by their respective neurons. They're also taken up by astrocytes who help with the recycling as part of their support role. The recycling depends on the citric acid cycle. It's an offshoot of the citric acid cycle. So we hypothesized that if we were getting significant changes in GABA, maybe we were seeing changes in the energy pathways of the cells that would eventually synthesize particularly GABA. And so moving into this area, we moved into looking at the energy pathways. And most of this work now is actually done in the fatal familial insomnia organoids. We don't have very much in the E200K, but I'll take you through what we have in the FFI. We undertook seahorse assay. So this measures mitochondrial function by measuring the oxygen consumption rate. And here we measure the baseline, then we inhibit the ATPase, which gives us ATP-linked respiration. FCCP allows free flow of the hydrogen ions across the membrane, which then allows us to look at the maximal respiration possible for that cell. And then we use rotenone and antimycin A to completely knock down electron transport, which gives us the background oxygen consumption that's unrelated to the mitochondria. And you can see all across the board here, the D178N FFI organoids have significantly increased function. We also saw that when we did a complex 5, the mitochondrial ATPase activity assay, and we additionally found that their membrane, their inner mitochondrial membrane, was hyperpolarized. You know, this supports that they're pumping the hydrogen ions very rapidly to keep that polarization and the energy production flowing. We looked at these under electron microscopy, SEM. What we found was that we saw quite a lot of mitochondria that were surrounded by this double membrane. It wasn't all of them, but there was a strikingly increased number of them. And Samote, then hypothesizing this might be mitophagy, looked at the autophagy induction ratio, which is an LC3-based assay. And indeed, it was up. It looked like there was increased autophagy, probably mitophagy in these cells. So we wanted to look at this a little bit in our CJD-infected organoids. It's a little bit harder to do some of these assays because we have to be careful how we handle the infection. It's very difficult to decontaminate, and of course it is infectious to humans. But we were able to measure the number of depolarized mitochondria using a fluorescent dye, and these are individual organoids. You can see that there's really no difference between the type 1s, the type 2s, or the control. However, we then undertook another qRT-PCR panel of genes looking at mitochondrial function. And now we actually got quite a few more changes, mostly things that were decreased, but there was particularly changes within the mitochondrial dynamics of the fission and fusion pathway. Less so in the fission pathway, but certainly more so in the fusion. The mitofusion 1 and 2 were significantly decreased across all of our infections here. This is kind of interesting in the sense that when mitochondria need engulfing, they're damaged, and the cell wants to activate mitophagy, these proteins, these 3, are actually modulated at the protein level. They're turned over faster so that these sick mitochondria will not fuse and be protected from mitophagy. So in this decrease, we're probably looking at an inability for these mitochondria to fuse, and therefore an increase in mitophagy. So that was pretty exciting and fitting with the D17AN data. Samote also looked at the lipid droplets within these, and this is what the red stain is over here and the quantification here. And he found a significant decrease in the lipid droplets. And lipid droplets are storage vesicles for neutral lipids, and in times of heightened stress, mitochondria can switch to using these as an alternative energy source. And indeed, we saw a significant decrease in all the neutral lipid species across the board by lipidomics. So this kind of told us that, well, maybe the mitochondria are struggling to get enough substrate. Maybe they're needing to switch to using different substrate, but is there a reason why? And so we did metabolomics on the D17AN organoids, and immediately you can see this is sort of a pseudo heat map of the pathways, that we have the buildup of a number of intermediates, the number of blockages within these pathways, within glycolysis, the citric acid cycle, and the pentose phosphate pathway. We see an increase in acetyl-CoA, which is the precursor to acetylcholine, so that may also account for that being increased. We also see a significant decrease in glutamate and glutamine, which are precursors for GABA and glutamatergic signaling. And here we're hypothesizing that maybe we're losing these because the cells are undergoing oxidative stress and are trying to take these intermediates and use them to restore their redox buffering capacity, generating more GSH. And this would also fit with funneling into the pentose phosphate pathway, because this pathway is also involved in regenerating redox buffering. So looking in the FFI organoids at markers of oxidative stress, we do see an increased number of ROS-positive cells. This is a fluorescent dye, and we threshold negative to positive cells. We also, by Western blotting, looked at an oxidative stress defense cocktail. And you can see catalase and SOD2 are mitochondrial antioxidants, and they're either unchanged or decreased, which was surprising. SOD1 and thyrodoxin are cytosolic, and we saw a significant increase in both of those, and also in SOD3, which is extracellular. So this was indicating to us that we may have a cytosolic oxidative stress going on. And just for reference, smooth muscle actin is this band here. It's actually supposed to be a loading control, but it is increased in astrogliosis. So this is why we rely heavily on our total protein loading controls, because we can't trust many of the traditional loading controls. Looking at our infected organoids, we were able to do qPCR for two antioxidants, the cytosolic SOD1 and the mitochondrial SOD2. And the SOD2 really agreed that there was nothing going on. We've also looked at depleting SOD2 in transgenic mice, and that had no effect on disease. However, SOD1, although it's very borderline, is significantly decreased. And in mouse models, knocking out SOD1, prion disease progresses faster. So this may indicate an ability to respond to a cytosolic oxidative stress is pretty important for maintaining viability. I was particularly interested in this because this could link in with a loss of PRP function. And the reason I say this is because at the same time we've been doing this work, we were looking at PRP function in mouse models and in hippocampal slice cultures. So again, using multi-arrays, and we put the hippocampal slice culture over the top. What we did was we treated these slices with potassium superoxide, which mimics an extracellular to cytosolic insult, and mitoparaquat, which is a targeted mitochondrial oxidative stress insult. We looked at their response initially, and their response of the wild type slices was very dynamic. They initially downregulated their neurotransmission and then rebuilt their network when the insult had gone away. For the knockout cells, those that got the potassium superoxide extracellular insult, they didn't downregulate any network communication, but it declined over time and they were unable to rebuild it. So they lost control of the reaction. And this was associated with significant oxidative damage to the actin cytoskeleton that was breaking down. And this is our wild type and our PRP knockout. And this here is DMPO, which is a spin trap. It attacks the spin orbital of a radical at the exact time it's formed and binds to it covalently. It's highly immunogenic, so we can come in and detect it with an antibody, and then you can co-localize it to see where those antibodies are. And we were finding in the co-localization, they were all over the actin cytoskeleton, so very likely involved in the cytoskeletal breakdown. So our conclusion to that paper was that PRP could protect against electrophysiological dysfunction and cytoskeletal damage by modulating the early ROS response and keeping it under control. We hypothesize now maybe during prion disease, the loss of function of that normal protein as it's being consumed may also produce this phenotype. And in our mouse models, and this is the hippocampus again, and this is the RML strain of prions, we see even at the preclinical stage, we're losing some signal from the cytoskeletal proteins. By clinical onset, there's a lot of the cytoskeleton that's already broken down, even though these mice will actually live quite a while longer before they die. In our infected organoids, we saw something similar. So this is microtubule-associated protein 2, and this is what our normal brain homogenate organoid slices look like. We've got sort of nice processes. And this is an example of one of our MV2s, and you can see the morphology is substantially changed. We haven't seen this in every infection and every slice, but of course, when you're sectioning through, it's the luck of getting the right slice. But so we think that maybe PRP, the loss of PRP as it's consumed, could result in this phenotype of cytoskeletal breakdown. So just to sum up what we have done here in our organoid models, both our infected and our mutation models, we're seeing evidence of an increased cytosolic ROS. The outcome of this seems to be a blockage in standard energy pathways. And actually, ROS are able to inhibit a number of the enzymes in glycolysis, and that results in a funneling of those intermediates into the pentose phosphate pathway, where they will regenerate cellular buffering against reactive oxygen species. So this may be an almost deliberate effort on the cell's part that is going horribly wrong, as those intermediates are getting stuck. The outcome of this is we have decreased GABA, and of course, this is resulting in a shift in excitatory to inhibitory neurotransmission. Seems like the cell is also in need of more ATP and certainly in need of another substrate that it can digest. So it's switching to using lipid droplets and depleting those in its effort to have enough energy. And as a side thing, as more and more PRP depletes, then potentially this ROS can directly attack the cytoskeleton, causing a breakdown, which of course would then feed back to a breakdown in neurotransmission as the synapses retract. So just in the last couple of minutes, I'm going to tell you a little bit about something different we've been doing with the organoids. And we've been using them as a model to test therapeutic efficacy. In prion disease, again, we have these great mouse models, and many of our animal models have shown huge promise with some compounds that has never translated into humans. There's probably a number of reasons for that. Mice and humans are different, but also a lot of therapies, when we try them in humans, we really wait until it's a compassionate use. So it may be far too late to reverse or stop even those changes at that point in time. What we're kind of hoping is that the organoid model can sort of slot into the existing models as a bit of a bridge for understanding whether a compound that works well in mice might be able to work well in human tissue. To do this, Brad set up a couple of treatment paradigms. He did what was called the prophylactic paradigm, where he treated before infection, during infection, and after, and then followed how infection affected those organoids or how much it developed. And then he did a therapeutic paradigm where he waited until they were infected and then treated them, and then looked to see how the infection would develop. These paradigms kind of were designed to represent someone who may have a genetic mutation, who wants to take some prophylactic treatment to slow the onset of their disease, versus someone who might turn up in the clinic already symptomatic and need treating. The compound that he used was pentosan polysulfate. It's a quintessential anti-prion compound. In cell cultures, it's really good at actually knocking down infection or even clearing it. In humans, it has been tried, but it's unfortunately got to be injected straight into the ventricles of the brain, which makes it compassionate grounds kind of use. And most of the studies indicated the patients didn't recover any quality of life. Some may have lived longer than expected. However, we don't have a blood-brain barrier, so we can treat the organoids directly. And this is a summary of the therapeutic data that we got. So this is the seeding activity, the number of prion seeds, again, indicative of the infectious units. And you can see after treatment, that's significantly decreased. Also, when prions misfold, they become highly resistant to protease digestion. So we can use that digestion and then Western blotting to look for their presence. And this is what it looks like when they're there. This is what it looks like after treatment. So this was pretty exciting that we had actually knocked down the replicating prions to a lower level. Incidentally, they did start to come back up without continued treatment. However, I think in prion disease, if someone was being treated, the likelihood is that they would be treated for the whole of the rest of their life. The one thing we didn't see with the pentosan polysulfate treatment was any improvement in their electrophysiological parameters. The infected organoids stayed flatly down on their electrophysiological signaling. Interestingly enough, the uninfected organoids actually got a boost in signaling as a result of the PPS. We don't know why that is, but it certainly was in stark contrast to the infected organoids. So we did hypothesize that if we want a therapy that's actually going to be particularly useful, we need something that could restore function as well as just knocking down infection. And so Katie decided to develop this paradigm for treatment of seeding neural progenitors back into infected organoids and looking at how this affected the disease parameters themselves, but also then the neuroelectrophysiological signaling. There is precedent for this. In Parkinson's disease, they have already started doing neural grafts into the brain. The first case study that I read was just one person, and they claimed that it did improve their function. So this is not without precedent. But she grew them out and infected them. She let them infect for 90 days when they're really fairly heavily infected. And at the same time, she differentiated from our iPSCs these monocultures of neural progenitors that she then seeded into the organoids. We only saw minimal changes in the disease parameters, a tiny little bit less seeding, a tiny little bit, one or two less organoids with deposition. But she did see a small but significant increase in the neuroelectrophysiology, indicating that we can measure this using this organoid model. And we hope that eventually it will be able to tell us whether a compound or a series of compounds can both knock disease down and help produce some functional quality of life for a patient. And so I'm just going to wrap up there. So in conclusion, we're excited that our model does faithfully represent the CJD subtype prions that we are infecting the organoids with. We seem to have an imbalance in neurotransmission in all of the organoids, which seems to involve a shift in excitatory and inhibitory neurotransmission, particularly linked with GABAergic signaling. Now, during CJD, the GABAergic population is one of the first neuronal subsets to drop out. So that could be interesting as a vulnerable population. However, in FFI, that population is less affected. So it's still a lot of investigation to go to understand what's going on. And we seem to have an over-requirement on the energy pathways that seems to be causing mitophagy. I would think that's downstream from some of the early problems of the prions, but certainly anything that damages the mitochondria is going to have a problematic influence on a cell. And we believe that we can use the organoids to look at therapeutic efficacy. In terms of our future directions, we are really interested in these subtype differences and how they're affecting the different brain cells in the different regions. So we have, with the help of the CJD Foundation, undertaken a big single cell where we've been differentiating organoids to represent more precise brain regions. They're directed to become more like certain parts of the brain. Then we've infected those, and we've done single cell sequence to see if we can tease out what cell types are affected by what subtype, and if it's the same or different pathways that become activated. Obviously, the cellular metabolism we're seeing a lot of changes in, so we've been investigating various aspects of cellular metabolism and proteostasis. Proteasomal function was linked with prion diseases a long time ago, obviously autophagy also, and in our studies. So we have some nice models of those now in the organoids, using mutations in various of those pathways to see how infection changes when we model that in organoids. We want to look a bit further at this GABAergic dysfunction, because a loss of inhibitory neurotransmission certainly could be leading to excitotoxicity. I'm still very interested in the role of PRP in cytoskeletal breakdown. If it's a loss of function, then potentially it could apply to all prion diseases. I don't think this will be the only avenue, though, because a loss of function certainly doesn't account for how those different brain regions are affected by the different prions. We are still doing model development. I mean, this has been a long model to develop, but there's a lot more things that we can look at. We have recently discovered that we don't need to infect for 7 days. We can now infect just for 24 hours, and we can infect with a much lower titer of prions than we thought would be possible. We're also looking, we were able to get fibroblasts from patients who died of CJD. And these fibroblasts have active propagating prions within them. So we're looking at reprogramming those into organoids and neurons to see how it differs, an infection that's already there from an infection that we start by our overlay. And we have a number of therapeutic testing avenues that we're also following. So I will just wrap up there. There's a lot of people that help in this research. Jay Carroll did an awful lot of the QRT-PCR for us. Brent takes care of all of our animals. Benji has been an absolute whiz with metabolomics and lipidomics. Most of our cells have come from Wenquan, who has now moved back to China, and our brain tissues from Jiangxi. And I'll leave it there. Thank you very much for listening.